all of the hard work that you're doing, what it goes to. And it's really been an explosion of knowledge in the last 10, 15 years at the basic science level that has been quickly translated into new approaches for treating leukemias and lymphomas. And I want to give you some sense of that with a concept called targeted therapy. The idea is that if you can identify something unique about a cancer cell, something, a protein on the surface of the cell, or a protein or a gene inside the cell, that allows you to develop a treatment that targets that protein or that gene and kills off the cells. That's the idea. And at the same time, leaves the healthy cells alone. And that's the ideal. And so there are several approaches that are being developed. The first is what's called targeted therapy against tyrosine kinases. I don't know if that word means anything to people. Certainly for CML, that's really the paradigm of where this started. In CML, there's a unique gene called the BCR-ABL gene that causes the whole process, and that produces a protein, the BCR-ABL protein, that's a tyrosine kinase. And it turns out that many kinds of leukemias and cancers are driven by these tyrosine kinase proteins. And it lends itself to developing drugs pills that can be taken by mouth that will target that tyrosine kinase and leave all of the normal cells alone. And so CML has been the real dramatic story with Gleevec, which was the first example in 2001. There are now 4 other tyrosine kinase inhibitors, or so-called TKIs, that are used for CML, and also in other kinds of diseases that are driven by tyrosine kinase as well. So that's one whole concept which has made a dramatic difference in the lives of patients with CML. When I first came to City of Hope, the only treatment that we would consider was a bone marrow transplant for patients with CML, and that was one of the most common diagnoses that would come to us for transplant. Now it's very rare for us to do transplants for CML because of how successful these oral agents are, and it's just made a dramatic difference. So that's one approach. The other is something called BITE, which stands for bifunctional T-cell engager, BITE. And what that means is you take 2 antibody fragments, and they're sort of created to be fused together. One of them binds to T cells, the cells in our body that are available to be activated to fight off cancer cells. And the other antibody binds to a protein on the surface of a cell, of a leukemia cell or a lymphoma cell. And so what happens with this small molecule, it brings together the tumor cell and the T cell brought right together, and that allows the T cell to do its job of killing off the cancer cell. And when that happens, then that antibody disengages and is able to go find other T cells and other cancer cells and kill them off. So that's a very new approach. The first one was FDA approved about a month ago, called blinatumomab, which is a mouthful. But the concept is really a terrific one because it can be adapted to almost any kind of tumor cell. As long as you can find a protein that's fairly unique to that tumor cell, you can design an antibody that will attack it in that fashion. So that's BITEs. And then the third approach is what's called CAR T cells. And that's something that's been in the news a lot. I don't know if anyone has heard about that before. CAR T cells. You'll be hearing a lot more. So CAR, C-A-R, that stands for chimeric antigen receptor. And the idea there is that T cells from the patient are removed from the patient, and they're genetically engineered to express a novel molecule that on the outside has a receptor against a protein on the leukemia cell or on the lymphoma cell. For example, CD19 is a protein on a lot of ALL and lymphoma cells. But that's the first one that's been studied, but there are other tumors that have different proteins that can be approached the same way. So on the outside of this T cell now, you have the receptor that will engage that protein, and then it's attached on the inside to a molecule that activates the T cell. So it makes it a very aggressive killing cell. And again, similar to BITE, this T cell now that has a receptor is able to find the leukemia or the lymphoma cells, bind to it, kill it, and then disengage, and then go find other cells. And so one T cell that's been engineered like that can kill up to 70,000 other tumor cells, for example. So that's been a very exciting development. And as I said, as long as there's a protein that can be identified that's either unique to the tumor cell or relatively so, and that there aren't other normal cells that have it, you can design this kind of a weapon to attack it. And so it's a very exciting time in the field from the basic science point of view, from the translational research, and clinically what we're able to accomplish. And it really depends on the support of all of you and people all over the country to support the research that's done at the basic science level, and then quickly get that translated into new therapies that make a huge difference. So I thank you all for all the work that you do. It helps all of us do the work that we do. And I'd be happy to answer questions if you have. Thank you.
Thank you, Dr. Snyder. I learned a lot.